Hello, everybody. This is Nafrit Sekho, and I welcome you on this platform, The Layers of Life Sciences, where we are discussing various aspects of life sciences in quite depth and detail, layer by layer. The objective is not just to help you in your graduation, post-graduation, and competitive exams, including CSIR NET, but also to have a general discussion about the life processes happening around us. In the first 6 videos uploaded on this platform, we have tried to discuss the structures of monosaccharides in quite detail. We know that just like other biomolecules, carbohydrates are also composed of basic subunits, which are these monosaccharides. To make the larger and more complex carbohydrates, these monosaccharides get linked to one another to form branched or unbranched chains. If these chains are composed of less than 20 monosaccharide units, then that carbohydrate will be referred as oligosaccharide. But if these branched or unbranched chains are composed of more number of monosaccharides, and the number can go till hundreds, then those carbohydrates are referred as polysaccharides. So we need to understand that most important phenomenon to make the larger and more complex carbohydrates is the linkage between two monosaccharide units, and then that chain of those two monosaccharide units may get linked to the third monosaccharide, and so on. So this linkage is really important. So let's start with the structure of oligosaccharides now. All the higher carbohydrates are composed of monosaccharide units linked to one another, as you can see over here. So these are the monosaccharide units, and these monosaccharide units are linked to one another through a linkage. Now, what this linkage is called? Whenever two monosaccharides are bonded with one another, then the bond between the linkage between them is called glycosidic bond. So you can see that if this monosaccharide is bonded to this monosaccharide, then this linkage between these two monosaccharides is known as glycosidic bond. So to summarize the things, it is that to form the higher, more complex and larger carbohydrates, monosaccharide units get linked with one another through the glycosidic bonds. Now, to understand the structure of these higher, more complex, larger carbohydrates, we need to understand the nature of this glycosidic bond, it means, right? Certainly, it is not a simple line between two monosaccharide units. We need to know how this bond is formed between two independent monosaccharides and how it is formed and why it is formed. So to know all the details, again, we will have to step into the realm of chemistry. So let's do that and try to understand this. So if we try to understand this one, we have done a part of it in the 2 videos, that is Biochemistry 3 and Biochemistry 4, where we learned the cyclization of linear structure of aldoses and the cyclization of the linear structure of ketoses respectively. So there also we studied that whenever any alcohol interacts with any aldehyde, then this alcohol leads to the electromeric effect in the carbonyl group of this aldehyde. So due to which the electrons of one of the bonds of the double bond between carbon and oxygen get shifted towards the oxygen, leading to the negative charge on oxygen and the positive charge on the carbon. So we can see over here. So over here, there is a negative charge and then the creation of the positive charge on the carbon. Within this alcohol, the hydrolytic cleavage of the bond between oxygen and hydrogen takes place, due to which both the electrons forming this single bond get shifted towards the oxygen, giving the negative charge to the oxygen and positive charge to the hydrogen. After that, simply the simple law of nature follows, which says that opposites attract. So this hydrogen gets attracted towards this oxygen over here and forms a bond with it, and this negative OR group makes a nucleophilic attack on this positive carbon. It gets attracted towards and forms a bond with it. So after whole of this hemiacetal is formed, where this OH is a new bond. So this electron shifted, so there is a single bond, and due to which this OH group is gets associated on one side. On other side, this newly added OR2 group gets associated, while its original two groups, that is alkyl group on one side and the hydrogen group on the other side, stay intact. So this is called the hemiacetal group, where carbon is associated with four different groups to fulfill its four valencies. On one side, it is associated with one alkyl group directly, while it is associated with another alkyl group indirectly through a ether group. That is, which means through an oxygen atom, it is associated with another alkyl, while on other two sides, it is associated with hydroxyl group and the hydrogen group, respectively. And this whole structure is called hemiacetal. This carbon, which was originally non-chiral, becomes chiral because over here it was satisfying two of its valencies with the help of same atom of oxygen, but over here it has got the chance to fulfill all its four valencies with the help of four different groups. So that's why this carbon becomes chiral, and this carbon is also called hemiacetal carbon, and this carbon is also called anomeric carbon when this carbon is the part of any monosaccharide, right? So till this part we did during the cyclization of the aldoses. Same thing happens when the ketone interacts with an alcohol molecule, and in there also this carbonyl group undergoes electromeric effect. This O gets ultimately associated with this H, and this OR directly gets associated with the carbon on the fourth side, due to which the structure which is formed is called hemiketal. Hemiketal is the structure where the carbon is associated with two alkyl groups directly and with the third alkyl group with the help of an ether group, which means that through an oxygen atom it gets associated with another alkyl group, and on the fourth side it is associated with the OH group. So the only difference between the hemiacetal and hemiketal is that over here on the fourth side, this is here there is a H, while this H because it is formed from ketone, so there is no H around the ketone, so therefore it is over here there is another alkyl group which is there. So this is what hemiacetal and hemiketal is. So this structure is certainly formed if one molecule is acting as the aldehyde group and another molecule is acting as alcohol. But if both the carbonyl group as well as the alcoholic group both coexist within the same molecule, then certainly still they interact with one another, same kind of electromeric effect is created. Similarly, hemiacetal or hemiketal is formed, and this leads to the intramolecular reaction between the two, leading to the cyclization of that molecule. And this is what happens during the cyclization of aldoses, which are basically polyhydroxyaldehydes, which means hydroxyl group and aldehydic group, or we can say carbonyl group, is present within the same molecule, due to which these any of the hydroxyl groups and the carbonyl group interact with one another intramolecularly and leads to the cyclization of the monosaccharides. And same thing happens within the ketones as well. There the ketoses get cyclized. This thing we have done in detail in the videos Biochemistry 3 and Biochemistry 4. Formation of the glycosidic bonds between these cyclized monosaccharides. So first of all, monosaccharide needs to get cyclized, and once it gets cyclized by the same process which I've explained over here, this is just kind of revisiting. So once they get cyclized, they are ready to get linked with one another through the glycosidic bond. So the process of formation of the glycosidic bond happens after the cyclization. How it takes place? It is that whenever, so hemiacetal is formed during the cyclization of aldoses, and hemiketal is formed during the cyclization of the ketoses. Now, when these cyclized hemiacetals and hemiketals again interact with another alcohol, and over here what happens is this alcohol once again ditches its hydrogen, right? And over here this carbon ditches its hydroxyl group, whole of this hydroxyl group. So the alcohol ditches its hydrogen, and this hemiacetal, or over here this hemiketal, ditches this hydroxyl group, and after ditching it gets associated, or we can say forms a new bond with this OR group, which means another ether group gets associated with this carbon. One ether group is already associated with this carbon, right, which happened during the cyclization. Now there is the formation of another ether bond. So over here you can see original alkyl group is as it is over there, then original hydrogen group stays over there, and now this is the ether group, or we can say this is the alkyl group to which it got associated during the cyclization process, or we can say in the last, when it interacted with the first alcohol. When it interacted with the first alcohol, this OR group got associated with it. Now when second time the hemiacetal interacted with another alcohol, now this time another ether groups gets associated with it. If you want to see the difference between the hemiacetal and acetal, it is that hemiacetal is associated with two alkyl group, with one directly, with another indirectly, while our acetal is associated with three alkyl group, with one directly and with two indirectly through the ether formation. We can say through an oxygen it is associated with two another alkyl groups, and on the fourth side it is still associated with the hydrogen, and over here this hydroxyl group has been replaced, right? And now if you will check over here, it has ditched this H positive, and over here this OH negative has been ditched. H positive plus OH negative will certainly form this water over here. Same thing will happen when the hemiketal is in the picture, which means it may be some kind of furanose ring of fructose or something like that. But when first time it interacted with the alcohol, it formed the hemiketal, and ether bond got associated with it. But now when the second time it interacts with an alcohol, then again this another ether bond gets associated with it. So if we compare acetal and ketal, you can see that ketal is associated with four alkyl groups, with two directly and with another two indirectly through the through an oxygen atom, that is through an ether bond. So, and certainly this H positive dish from here and OH negative coming from here, H plus OH forms the extra water. And this reaction is actually the basis of the formation of the glycosidic bond between the two monosaccharide units. Those monosaccharide units may be same or they may be different. They both may be aldoses. They both may be ketoses. It can be anything, but glycosidic bond is formed in the similar manner. Actually, this carbon, which you can see over here, is actually it is the hemiacetal carbon. This is actually the carbon number 1 in case of aldoses. And if I am referring to this carbon in the case of ketones, this is the carbon number 2 in the case of ketosis. We know that on the carbon number 1 of all the aldoses, the carbonyl group is present. So this carbonyl group certainly gets replaced with this OH and OR. Then again on the same carbon number 1, when interacts with this group, then another OR. This C is nothing but the carbon number 1 of first monosaccharide unit, and this R is nothing but another monosaccharide unit. There is the continuation of the similar kind of reactions, that is stepwise step formation with the action of aldehyde and alcohol together. Hemiacetal is formed, and then progressively when again there is an interaction with another alcohol, then again acetal is formed. Similarly, with the interaction of ketone and alcohol, hemiketal is formed, and progressively when another alcohol interacts with that hemiketal, then ketal is formed. So there is just a sequential reaction basically over here. But the major difference is that when this hemiketal, when this aldehyde and alcohol interact with one another, at the first place they are present within the same molecule, and this reaction where the hemiacetal and hemiketal is formed during the cyclization of aldoses and ketoses, that was an intramolecular reaction. But when this hemiacetal again interacts with an alcohol, this time this is gonna be the reaction between two different molecules, which means that this reaction is gonna be an intermolecular reaction. This R over here is nothing but this R3 is nothing but whole of the another monosaccharide unit, and this OH is nothing but one of the hydroxyl groups present in that monosaccharide unit, which will interact with the carbon number 1 of the first monosaccharide unit. So this is how this is gonna happen, right? And over here also same thing will happen. This R4 is nothing but another monosaccharide unit, and this carbon number 2 is nothing but the carbon, the anomeric carbon of the first monosaccharide unit. So this is the carbon of first monosaccharide unit, then oxygen bonding, the oxygen in the middle, right? It is kind of the middleman. So, and this is the another monosaccharide unit, and this middleman, this bond, right? The oxygen, this bond, and this bond, oxygen, and this bond. This is basically the glycosidic bond. So let's see how it happens. So we know for the formation of the glycosidic bond, we need the first thing is the hemiacetal, and another thing is alcohol. So whenever hemiacetal and alcohol will interact with one another, then glycosidic bond will be formed. If I say the molecule which is gonna act as hemiacetal is the glucose, and the molecule which is gonna act as this alcohol over here is again the glucose. So the molecule which is going to act as hemiacetal is this glucose unit, and the one which is going to act as alcohol is again the glucose molecule. So actually, if we try to understand, it is the role play. Okay, both of the molecules which are going to form the glycosidic bond among themselves, both of them are capable of acting as hemiacetal as well as alcohol. It is just the way they decide. The the glucose unit, which is gonna decide that, okay, I will play the role of hemiacetal, it needs to make available its carbon number 1, right? That is the first thing. Second thing, it should be ready to lose that hydroxyl group present on the carbon number 1. So you can see, if we go back over here on the last slide again, so this hydroxyl group is lost, so this carbon number 1 should be ready to lose its hydroxyl group. So same is the case over here. And if some glucose wants to play the role of alcohol, then first of all, there is no restriction of availability of specific carbon, right? Carbon number 1, 2, 3, 4, or 6, apart from 5. Rest any carbon can be participating in this bond formation, no restriction, while the glucose or the monosaccharide, which is playing the role of hemiacetal, should make the carbon number 1, or if it is the aldoses sugar, or carbon number 2 if it is a ketoses sugar, which means that they should make their anomeric carbon, or we can say hemiacetal or hemiketal carbon, available for the participation in the glycosidic bond, and that carbon should be ready to lose its hydroxyl group, right? But in the case of the monosaccharide, which is gonna play the role of alcohol over here, no restriction, any carbon can actually participate. Carbon number 5 cannot participate because it has no hydroxyl group. This all the carbons which are having the hydroxyl groups, any of them can participate, first of all. Secondly, that carbon, whichever carbon is gonna participate, that carbon should be ready to lose its hydrogen from its hydroxyl group. So they all have the hydroxyl groups, but the carbon which is ready to lose its hydrogen from the hydroxyl group, that carbon can participate. So this is the one which is playing the role of alcohol. So over here you can see that both of these are the glucose units. Yes, some variation is there regarding the stereoisomerism. This is the alpha glucose, right, and this is the beta glucose, but that doesn't make any difference. Even beta glucose can also act as hemiacetal. This stereoisomerism does not interfere with their capability to act as hemiacetal or act as alcohol. So what happens now? So this glucose is acting as the hemiacetal, and this glucose is acting as the alcohol. So let's try to find the similarities between these two molecules. So let's see, I'm talking about this carbon number 1. Hemiacetal carbon is this carbon number 1, right? So this over here, this carbon is the carbon number 1, right? So this is the carbon number 1, because this is basically aldose. If it would have been some kind of ketose, be it fructose or any other, right, and it would have been a furanose ring, then certainly I would have been referring to the carbon number 2 over here. Right now over here, this is the carbon number 1. This carbon is associated with hydroxyl group on one side and the hydrogen group on the other side, right? While on the third side, it is associated directly with the alkyl group. So whole of this is the carbon right over here. So whole of this group is actually the alkyl group till the carbon number 6. This is the carbon number 6. So this is the alkyl group with which it is associated directly. And then to the second alkyl group, it should be associated with the help of or through an oxygen atom. So this carbon over here, then this is the oxygen, and this is the alkyl group to which it is associated through an oxygen atom, or we can say this is the ether bond, the first ether bond, which is already existing over there. This is about the hemiacetal carbon. Now let's go to this one. So OH group. Now over here, I have told you that any of these carbons which are having the hydroxyl group can participate in this bond formation. We are choosing the most common carbon which actually participates, which is generally the carbon number 4. So if this is the carbon number 1, 2, 3, and this is the carbon number 4. So this carbon number 4 is this OH over here is actually this OH, and Then after this, everything, whole of this part, right? Whole of this part is basically the R, is basically the big R. Okay, so this is OR, OH, R, 3, you can say R3. So basically now what needs to happen, that this OH from here needs to be replaced with this OR. So this OH will be lost and this H will be lost, and this OR will come and join itself on the fourth site. Okay, on the fourth site, and will convert this hemiacetyl carbon to the acetyl carbon. So they are gonna interact with one another. OH from here and H from here are actually lost. They have been removed. Each of these glucose molecules have played their part well. And now what is gonna happen next? The next thing is that there should be the bond formation between this and this, right? And this C over here, there is a carbon, there is a carbon over here, this C, and this bond O, and there is another carbon over here, this COC, there is ROR bonding, is another ether bonding, and this COC bond, when this ether bond, when it is formed between two monosaccharide units, this bond is called glycosidic bond. Let's see how it happens. So in this way, there that bond COC bond has been formed between the carbon number 1. This carbon number 1, earlier it was referred as hemiacetal, now it has become acetyl. With the at one position it is associated with directly with the first alkyl group, and then it is associated with one hydrogen, right? While at two positions, it is associated with the two alkyl groups through the oxygen. The first is this one, CO, and then whole of this alkyl group, while the another one is CO and whole of this alkyl group, right? In this way now, because now it is associated with the two ether groups, as it happens in the acetyl we have done in the earlier slides, that's why now it is called acetyl carbon, not hemiacetyl carbon, right? Now, this structure where there is a bond between the carbon number 1 of one glucose unit and the carbon number 4 of the another glucose unit, and where the first one was the alpha glucose while the second one is the beta glucose, then if a bond is formed like that, so in short I want to write their names, I will write as this one is glucose, right? That is this is the short form, that how you can learn. So this is a glucose. This glucose first carbon is participating in the bond, right? So after glucose, I need to write about this bond, right? I'm just trying to explain this bond. So this glucose bond is between the alpha. So this carbon was alpha 1, right? So there is alpha 1, and then there is this 4, right? Carbon number 4. This bond is between carbon alpha 1 carbon and 4 fourth carbon of the glucose, which is basically in the beta conformation. So over here I will write beta, and what is the unit? 4 beta is what? Another glucose. So this is one way of writing the name, right? This is the short form which helped me to learn the structures of so many oligosaccharides, basically the disaccharides. So this is the first disaccharide which we have come across, and its name is maltose. And you can see that, okay, if the hydroxyl group has been lost from here and H has been lost from here, so a water molecule has been formed at the end, right? So over here, so this is the maltose where one glucose unit forms the bond with the another glucose unit, and the carbons which are involved are carbon number 1 of the first glucose unit and carbon number 4 of the second unit, and the second carbon unit needs to be in the beta conformation. Okay, that much is clear. So maltose is nothing but it is a maltose is a disaccharide which is actually found in malt. Malt is actually a grain which is used for brewing the beer and all that, and it is also found in large quantities in all the germinating seeds. And its structure is that two glucose units are connected with one another through a glycosidic bond, and the nature of glycosidic bond is that carbon number 1 and carbon number 4 are actually involved of those two glucose units. Now, if you will actually see, this carbon, which was actually hemiacetal, has lost its hydroxyl group, right? And now it has become acetal. But the hemiacetal of the second glucose unit, right, is still there. It is still intact. So now if some another alcohol will come, then it can easily get associated with it. It will readily lose this hydroxyl group. It will readily ditch this hydroxyl group to get associated with another monosaccharide through that ether linkage. Whenever there is free hemiacetal for the further reactions, we say that this sugar has the reducing end free. This is the reducing end of this sugar, right? Why this is called reducing end, I will explain that in the next video. Basically, over here this is the non-reducing end of the sugar, because over here there is no hemiacetal. If anomeric carbon is there, which means that if hemiacetal or hemiketal carbon is there, not associated with anything, it is free, right? Then that end is the reducing end, and the end where there is no anomeric carbon, that end is called non-reducing end. So this end of this disaccharide unit is non-reducing end, and this one is the reducing end. Any sugar, any oligosaccharide, any disaccharide may or may not have the reducing end. Over here, reducing end is there. Right? We will study about the cases where there is no reducing end, right? We will go through those cases as well. Now, this is the short form of learning the structure of maltose. If I want to learn the structure of maltose, I will say, okay, the two glucose units, alpha 1 4 beta linkage. Oh, this is how I will learn it. Now, if I want to write the scientific name of this molecule, then how I will write. So over there, I will have to mention each and everything, which we actually mentioned while we were doing the naming of the cyclic monosaccharides, right? We did the naming of the cyclic monosaccharides again in the videos Biochemistry 3 and 4 in extreme detail. Please go back, do watch those videos, and then come back to this portion. So this is the glucose. Which form of glucose? Alpha form. So we will write alpha, then D-glucose. This is D-glucose. How I know that this is D-glucose? Because carbon number 6 is pointing upwards. This is the first sign that you are dealing with a D-glucose. And again, 245, that code which you will learn in the video Biochemistry 2. Again, very important video for the detailed understanding of the monosaccharides. 245, fifth has already lost its hydroxyl group during the cyclization, right? So over here, the carbon number 2 and carbon number 4, both are having the hydroxyl groups downwards. So it means this is D-glucose. D, right? Gluco. Now, this ring is which one? Is it a furanose ring or pyranose ring? This is the pyranose ring. So I will write glucopyranose. Okay, pyranose, not pyranose. If I would have written the name of the glucose, the cyclic glucose, I would have written this name as alpha D glucopyranose. But here it is not pyranose. There is no hemiacetal carbon over there. Now that hemiacetal carbon has been converted into acetyl carbon, now I need to write alpha D glucopyranose. Okay, then which bond? 1 4, right? Pyranose 1,4 bond of between what? Beta D gluco, just the prefix gluco, again glucopyra nozzle, no, pyranose. Here amiacetyl carbon is still there, so I can easily write and comfortably write its name as beta D glucopyranose. So this is the way how do we write the scientific names of all these disaccharides. This is the disaccharides, and disaccharides are actually the most common oligosaccharides to be filled, right? So this is how we write the scientific names. This is the short name, and the general name is maltose. Okay, so we need to discuss few more disaccharides over here. So this first one is lactose. As you must be knowing that what is lactose? Lactose is basically the disaccharide sugar which is present in the milk and milk products. You must have heard about the term lactose intolerance, which is caused by the lack of production of an enzyme called lactase, which leads to the digestion of this lactose, right? So first of all, let's understand what this lactose is. First of all, lactose is composed of two monosaccharide units. Both are aldoses. One is galactose and then the second one is glucose. Galactose over here is acting as the source of hemiacetal. It is acting as hemiacetal, and glucose is acting as alcohol over here. And this bond formation takes place between this hemiacetal and this glucose, right? Now, this its carbon number, there is a bond between this carbon number 1 and the carbon number 4 of glucose, and that bond is basically beta beta, right? So both are in the beta forms. Galactose is also in the beta form and glucose is also in the beta form. That's why this is the common way that galactose, the short form of galactose, GAL, can be written over here, and then it is beta 1,4 glucose linkage, right? And this is its scientific name, beta D galactopyranose, right? Because this was galactose, and which galactose it is, you can see OH over here, which actually took part in the formation of this glycosidic bond, was directing upwards. So upward means beta. So beta D form. Why? Because sixth carbon is pointing upwards. If it would have been L form, then it would be present, it should be directing downwards, and it would be directing upwards. So this is D. So beta D galacto is its prefix for the word. We need to take that. We don't write galactose over here. We always drop O's, while whether we are discussing glucose over there, we use the word gluco. And so this is galactopyranose, right? Of course, this acetyl carbon, hemiacetyl carbon is not there. Now it is the acetyl carbon. So pyranose 1,4 beta D glucopyranose. So this is the same, the way we discussed the structure of maltose. Maltose was beta 1,4 bond between two glucose units, while lactose is the beta 1,4 bond between one galactose and one glucose. And moreover, both are here, both of them are beta, while in maltose, in the maltose, one of the glucose, the one which was acting as hemiacetal, was alpha, while other one was the beta. Now, after that, we need to go to the next disaccharide sugar, which is sucrose. Oh, sucrose. The villain everybody tells, don't eat sugar. This is actually that sugar only. So this sucrose is actually formed of glucose and fructose, two sweetest sugars. So that's why it's so sweet. And this is also called cane sugar. And the doctors are constantly telling to moderate the consumption of sugar, and all the fitness experts are regularly telling that totally cut it off from your daily routine. For me, it's really difficult. We need to study its structures. Two monosaccharide units which are involved is glucose, and another one is fructose. Fructose in the furanose form. the monosaccharide which acts as the hemiacetal is the glucose, and the monosaccharide which acts as a source of alcohol, or we can say acts as an alcohol, is fructose over here. And the bond is between, of course, the first carbon and the second carbon of the fructose. Now, if you see the structure of fructose, the carbon number 2 has been depicted on the left hand side. Generally, if somebody will tell me to draw the structure of fructose, what I will do, this is the oxygen at the top, this is the carbon over here, and there is certainly a carbon over here. Now, this is carbon number 2 over here, and to this carbon number 1 is certainly attached, which is not participating in the ring formation. And then fructose is basically a hexose, right? Ketohexose. So of course there are 6 carbons. So carbon number 1, 2, 3, 4, 5, and this is the carbon number 6. So carbon number 2 is always drawn conventionally towards the right hand side, not on the left hand side. But to show the bond formation over here, we had to flip the molecule, right? Whatever was on the right hand side, that we had to show on the left hand side. This is just the representation, right? So we had to flip. When we flip, now we know that the code for fructose is 45, right? This is basically 45. Right, on the carbon number 4 and 5. On 5 it is not possible because it has taken part in the formation of this ether, which led to the final cyclization. So basically on carbon number 4 it should be downwards, but if you would check over here, it has been shown upwards. This is just because the molecule has been flipped. So it is actually the rule that if anomeric carbon taking part in the bond formation, then we need to flip the monosaccharide, the structure of the monosaccharide, which is taking part in the bond formation as alcohol, right? So the second one is taking part as alcohol, and the carbon which is involved in the formation is basically the anomeric carbon. So over here we will have to flip that structure. If you are flipping the structure, you are not showing the anomeric carbon on the right hand side, rather it needs to be represented on the left hand side. Then the vertical flip also takes place. It's not just the horizontal flip of the things. The vertical flip also takes place on each and every carbon. The direction of the associated hydroxyl and hydrogen groups also get flipped. So on carbon number 4, if I'm showing over here where the carbon number 2 is shown on the right hand side, over here I will certainly show the carbon number hydroxyl group downwards, right, and this upwards, and on carbon number 3 it will be upwards and downwards. Why so? You will be able to understand if you would watch the videos Biochemistry 3 and 4 over here, but it's all flipped. The flip is just otherwise you would say this, whatever we have done till date in the earlier videos, it's all gone wrong. No, it's not gone wrong. It is just we have flipped the structure to show the bond between the two anomeric carbons right now. So that's why all the substituents on all the carbons have also been flipped vertically as well. So over here, this will always happen whenever the two anomeric carbons are actually forming the bonds among themselves. Then this flip will always happen with respect to the second monosaccharide, or we can say the monosaccharide which is taking part in this bond formation as an alcohol. This is basically, that is very important point over here. Now, general structure we can easily learn as that there is one glucose over here. Then after that there is alpha, that was in the alpha form, and it is the bond between 1 and second carbon of the another monosaccharide, that is fructose, and fructose was in the beta form. So even if you would say that, you would say no, it is not in beta form, it's in alpha form. It's like now when the molecule is flipped, whatever was the alpha form, it will become the beta form. It will be called the beta form only. So everything is flipped regarding this structure. right? So right now, yeah, this was in the beta form. So alpha beta bond, and between carbon number 1 of glucose and carbon number 2 of fructose, both of them are the anomeric carbons. Now, right now, both the anomeric carbons are actually involved in the bond formation. So this disaccharide, that is sucrose, has no reducing end. Both the ends are having non-anomeric carbons, right? So that's why these both the ends are basically the non-reducing ends. Why they are non-reducing ends, I will explain in the next video. Till then, it is just a mystery. So now we need to come to tree halos, which is very important disaccharide present in insects basically, but very simple one, because again it is composed of two glucose units, and those two glucose units are, one of them is acting as a hemiacetal. There it was mandatory for it to involve its carbon number 1, but the glucose unit which was acting as alcohol, it could have involved any, but to form trihalose, it involves its hydroxyl group present on the first carbon only. So this over here, this is the first carbon. So both are glucose units, and both are involving their first carbons. But if you would just compare the structures over here as well, so you would see that over here, this is, both are alpha, right? First of all, both are alpha. It means OH group should be pointing downwards, right? But over here, you would say it was pointing upwards, right? But still, it's not beta, because again, anomeric carbon is involved in the bond formation. The structure is flipped. Carbon number 1 is always shown on the right hand side. But right now, to show the bond formation between the two carbons, we need to show it on the left hand side. So everything needs to be flipped, right? So whatever is beta will become alpha. So over here, this alpha, both are alpha first of all, and this is a normal glucose unit. Carbon number 2 and carbon number 4, the hydroxyl group is present downwards. Carbon number 6, it is upwards. Right now, on this glucose, everything is flipped. Whatever is upwards on first glucose, on the second glucose, after getting flipped to form the bond, everything has been flipped horizontally as well as vertically. So carbon number 6 is downwards. Carbon hydroxyl on carbon number 4 is upwards, although it should be downwards. On carbon number 3, it should be upwards, it is shown downwards. On carbon number 2, it should be downwards, it is shown upwards. So everything is flipped. So both are glucose units. You would still find the difference in the structure. There is no difference in the structure. It is the difference in the representation, just right. So here the structure is flipped. So that is the important point. One more important point, we did scientific names of these disaccharides, right? So if you would read that till this, it is alpha D gluco. The structure is pyranose, so pyranosyl, because now it is the acetyl carbon right now. Beta, then there is actually 1 2 here. It should be written 1 2. It is missing. So 1 2 beta D fructo furanose. It should have been a furanose, but it is not furanose, because again this has also lost its anomeric carbon, right? No anomeric carbon over here. So whenever the second monosaccharide unit also involves its anomeric carbon in the bond formation, then it does not end with the word nose, which generally happens. It will end as side. Fructofuranoside, furanoside, glucopyranoside. Here also the carbon number 1 is involved, right? So there is no hemiacetal carbon. Here there is no hemiketal carbon. If it would have if the hemiketal carbon would have been free, then we would have named it as fructofuranose, right? But over here that has not happened. So all the sugars, or we can say all the disaccharides, if we need to write their names, then we need to add the word "ide" in the end to denote that it has non-reducing ends only, right? No reducing ends. So this is about the disaccharides. Apart from disaccharides, we come across many other oligosaccharides also. So the most important ones which we come across are basically the fructo-oligosaccharides and galacto-oligosaccharides. Sometimes there are gluco-oligosaccharides as well. So what are these fructo-oligosaccharides? These are actually the sugars which are added, and they have low glycemic index, they have sweet taste, and they act as prebiotics because they don't get actually digested. They act as fibers rather than acting as sugars in our bodies. They act as fibers, they act as prebiotics because they are not being absorbed by our small intestine, and in fact the good bacteria in our gut feeds on them. That's why they act as prebiotics. So what is the structure? If I just go by the simple structure, it is that the first position there is glucose. This glucose is associated with the fructose units, right? First fructose, and then the fructose unit forms the chain of 3 to 10 units, and one fructose being associated with another fructose, and suddenly the bond formation between all these monosaccharides, this bond between all these monosaccharides is which one? It is the glycosidic bond. And now, if we go into the details, then we need to know that which kind of glycosidic bond. Now we know that there are so many combinations possible, right? So how it happens basically. So if you would see that the first unit is always glucose. So this is the pyranose ring of the glucose. Now over here, this glucose is in the alpha form. So because this is in the alpha form, hydroxyl over here is lost, right? And this fructose, the first over here, this is acting as the hemiacetal, right? So this is have this glucose, the carbon number 1 of this is acting as the hemiacetal, and this first fructose will act as the alcohol, and it gets involved. It's hydroxyl group present on carbon number 6. So this is carbon number 2, this is 3, this is 4, this is 5. And if you say this is the carbon number 6, and suddenly with this carbon number 6, there would have been an H. Now this hydroxyl group gets involved over here. This H is lost. This H is lost, and from here OH has been lost, and a bond is formed between this carbon and this carbon number 6 with the help of this oxygen, which was actually the part of the hydroxyl group associated with the carbon number 6. So this bond is basically alpha. So this is alpha, and which carbon number 1? Certainly carbon alpha 1, right? Glucose alpha 1, and this bond is alpha 1 and 6. Now which form is there of fructose? Beta. 6 beta. So it is 1,6 glycosidic bond, right? So in short, we generally say it's 1,6 glycosidic bond. Now, once this bond is formed, then this fructose again acts as an alcohol. So it always acts an alcohol. Over here also, during this bond formation, it acted as alcohol. So this is a permanent alcohol, fructose. So this acts as an alcohol, and with the help of this carbon number 1. So this is carbon number 2, this is carbon number 1. There must be a hydroxyl group over here. And now when this hydroxyl group attacks, this carbon number 2 loses its hydroxyl group, which was there in the beta position over here. So it loses its hydroxyl group, and a bond between the carbon number 1 of this first fructose and the carbon number 2 of the next fructose is formed, right? So this kind of formation, so all of them are in the beta form. So this kind of formation is basically 2,1, right? So this is beta 2,1 bond. Now this is this unit, fructose number 2 has been associated. Now this fructose number 2, on this fructose number 2, again, there is certainly on the second carbon, carbon number 1 must be again associated with it. Now this carbon number 1, which is originally having OH group, again acts on this carbon number 2 of the next fructose and forms a bond with it. So again, the same bond, beta 2-1 bond is formed, and this process continues. Now this hydroxyl, again this is carbon number 1, and there is a hydroxyl group present over here. This hydroxyl group will again attack as an alcohol on the carbon number 2 of next fructose, which will act as the hemiketal, right? So that will act as a hemiketal. So over there again, same process will be repeated, and this glycosidic bonds will 2-1 kind of glycosidic bonds will keep on forming till there are 3 or 4 or 5 or 6 or 10 units are actually get associated in form of a chain. In this way, this oligosaccharide chain is formed. Similar structure is there with another well-known oligosaccharide, which is basically galacto-oligosaccharide. So this is the galacto-oligosaccharide. Over here also, the first unit is always glucose, which acts as a first hemi-acetal. When then after that, the galactose forms the bonds among themselves only. And these bonds are so varied. Sometimes they form beta-1,3, sometimes they form beta-1,4, sometimes those bonds are beta-1,6. It means there is variation. I cannot give one structure of galacto-oligosaccharides because this keep on varying. Even this first bond between glucose and first galactose, it may be beta-1,3 or beta-1,4. It can be any of them. So in this way, here we end our discussion about the oligosaccharides. But as we have come across the reducing and non-reducing ends, so we will have to wind up this with the discussion of reducing and non-reducing sugars. About that, we will discuss in detail in the next video. Till then, goodbye. Take good care of yourself. Keep revising the things. It helps. Thank you. Bye-bye.